exactly in the same way that we find that animal cells specialize through the process of differentiation, so do plant cells. So we have here the idea of specialization, in this case of plant cells, through differentiation. And differentiation is a super term which I encourage you to use as accurately and frequently as possible. So we've got the process of differentiation. Now, I want to immediately introduce you to some friends of ours called root hair cells. Root hair cells. Now, these root hair cells, they are differentiated in a way really to achieve two things. They're connected. And those two things are, first of all, to increase the surface area, of the root. Now, if we have a look at this magnified image we have of a root here, we can clearly see that this root branch has these root hair protrusions, and these root hairs increase the surface area of the root as a whole. So that's obviously really, really positive and a functional feature of this cell. Now, the second point I would make, leading very much on from that, is that as a result of this, they are capable of absorbing more water, typically through the process of osmosis, and dissolve minerals, which the plant the plant does achieve through the process of active transport, much more of which when we look at those things specifically in future lessons, just some terms for you to have for now. But these are effectively our 1, 2, and the functional reasons why our root hair cells differentiate in the way they do. So that being understood, let's see if we can actually look at the features of this cell. Well, I guess the most obvious thing is to recognize that this here is the root hair. Fairly obvious, but let's make sure that we label that in there. Secondly, notice some of the features which we typically expect to be in plant cells are also present here. So let's just go through one or two of them. We've got a cell wall, okay, cellulose based. We have a cell wall. We have a nucleus, which is controlling the activity of the cell in here. We also have mitochondria, numerous of them, and the mitochondria are here and here and elsewhere in the cell as well, of course. And we also, let's go for a darker color, we also have in evidence, we have our permanent vacuole. So all those are relatively familiar to us, and we can see those nice and clear. I should, of course, also mention the cytoplasm as well. Let's also make sure that we mention that too. So those features are what we'd expect. Now, one thing that is not present, and I'd encourage you to think perhaps, why that might be, is we do not have any evidence of chloroplasts. Now, if you consider that we have no chloroplasts and that this cell is underground, if we consider that chloroplasts are the site of photosynthesis, then of course, because there is no sunlight penetrating underground, certainly unless it's at a very, very shallow level, because there's no sunlight, therefore chloroplasts are kind of irrelevant to this plant, to this root hair cell. They're not pleasant. Now, a couple of additional things I just want to remind you of. The mitochondria, they are the main site of aerobic respiration, where we get the release of energy. And of course, the root hair cell, which we've said already, increases the surface area of that plant. So just to be super clear on this, the root hair cells are differentiated to perform their function. What is their function? To increase surface area and to absorb more water through osmosis and to more dissolved minerals through the process of active transport. Now we've got a good understanding of root hair cells. Let's have a look at another example of differentiation in plants. And I'd like to introduce you to what we have in the image here, which is a xylem vessel. Now notice I'm not using the phrase xylem cell there. Why? Because what we've got here in the illustration is actually several cells. So a kind of a nice way to kind of picture this is imagine that here this was once a cell, this here was once a cell, for example. And we can see immediately, let me just get rid of them for a second, we can see immediately that the end walls have broken down. So that as a characteristic in and of itself is kind of interesting. Why would these end walls break down? And if you look at just kind of the aesthetic or the visual structure of this vessel, and we're even referring to it as a vessel, you might start to realize what the function of this vessel is. And that's what I'd like to quickly confirm with you now. The function is to transport water and minerals, up the plant, and it is only up the plant, by the way. Okay, we're going to look at a structure in a second which transports substances up and down the plant, but we'll come back to that in a second. Now, a couple of features of this, as we've seen already, it's hollow, and interestingly, the cell is waterproof. Now, in order to achieve that waterproofing role, we need the presence of a chemical called lignin. Now, that, of course, immediately begs the question of what the heck is lignin? I will answer that in just a moment's time. Before we get to that, though, a couple of key things. These cells are found in the stem, the root, and the leaf. Okay, and the leaf. Okay, so this is not just sort of like the stem of the plant. Sometimes I've had students in the past who think, well, that's obviously how plant stems work. Well, yes, they do feature these cells, but these cells are also found in other features of the plant as well. Finally, before we look in more detail at lignin and its role, can I just emphasize to you what we don't have here? We don't have a nucleus, we don't have cytoplasm, we don't have chloroplast, and we don't have mitochondria. Now, that really leads us neatly to come back to this point about lignin then. So as I've said to you already, lignin is waterproof. So let me rephrase that. Lignin waterproofs the xylem vessel. Okay, it also makes the walls of the cell stronger. Okay, so we've got a waterproofing effect. We've got the fact that it makes the cell stronger, but it also kills the cell. So it makes the xylem vessel non-living. Okay, and that is ultimately why it is possible to have this cell type without things like the nucleus, for example. So we've got a non-living cell, but it is absolutely perfectly differentiated to perform its function, which of course is to conduct water and minerals up that cell. Okay, one kind of final point that I think is really nice to drive home here. It's a nice summary really, and it's something that if you can get into your answers in your exams, then that is a great thing. And it is as follows. As xylem differentiate, okay, so as they undertake the process of specialization through differentiation, so as they differentiate, the cell gains lignin. And it's this lignin that has this waterproofing, strengthening, and ultimately is what causes the cell to no longer be living once it is matured into its differentiated format. And as the final example of differentiation in plant cells, I want to have a look at phloem. Okay, phloem. Notice the spelling, please. Phloem. And the first point I would make to you is that phloem is a tissue made up of two different cell types. Now, this therefore becomes kind of interesting because those two cell types, if we name them first of all, the first one is what we refer to as sieve tubes. Have a look at the illustration and see if you think you can recognize where those sieve tubes are. And the second example, or the second type I should say, is what we call companion cells. Companion cells. And again, take a look at the illustration and see if your common sense and intuition suggests to you which feature are the companion cells. Okay, now I also want to talk to you about the function of phloem. Okay, so it's always useful to look at the function because then once we look at the structure of the cells, in this case the cells, we can see clearly why differentiation has occurred in the method in which it has. So the function of phloem is to carry dissolved sugars up and down the plant, up and down the plant. Notice the difference, therefore, between phloem and xylem, for example. Xylem only carries water up the plant. Our phloem can carry up and down these dissolved sugars, so it's worth just highlighting that as well. So let's have a look at this. Let's first of all have a look at our sieve tubes. Here we go. I'll sort of keep things in pink to match up here. Here, as an example, is just a broad outline of a sieve tube cell. Here is a second. Notice what these sieve tubes do not appear to have. They have very few mitochondria. They have no nucleus. They have little cytoplasm, and because of all that, one positive thing is they have little resistance. So again, to come back to the function there, they've got little resistance to the movement of sugars, to the movement of sugar. So that makes sense, bearing in mind our function. But a little like our xylem, because of the absence of these typical cell structures, such as a nucleus, for example, alone this cell would not be able to live. And that, of course, is where the companion cells come in. Now, the companion cells, they are connected by pores. That's to the sieve tubes, I mean. They have lots of mitochondria. Unlike the sieve tubes. And importantly, and really emphasize this statement, they can control the function of both cells. They can control the function of both cells. So we've got some pretty predictable cell features in the companion cells. We have our permanent vacuole, we can see our nucleus, we can see mitochondria, we have some example of ribosomes within the cell. But what we also kind of want to identify is two additional things here. We have our kind of pores, which allow communication with the cell, and also if we have a look at these kind of end walls of this time the sieve tubes, we can also recognize that the end walls feature what we would refer to as sieve plates. Okay, sieve plates. And it's those sieve plates, remember they were previously the end walls, bear in mind, they were previously the end walls. What they've done is they've differentiated to ultimately become sieves. You see these holes in here which help to support the main function of these cells, which is the carriage of dissolved sugars.